Today we are going to talk about development networks and look at a specific example of a network that leads to skin cells with spikes or without spikes in a thorny dragon. First off, a developmental network is a pattern that occurs in the DNA that determines whether or not a specific gene will be expressed due to the presence or absence of proteins on the strands. In our example, the network we will be looking at is for a gene in thorny dragon skin cells for spiky skin or non-spiky skin cells. The network is this. To break this down so it is better understood, S activates P represses I represses K activates E, and E activates the spike gene in the cells. If something is repressed, then it will not be expressed in the network and can't repress or activate anything downstream. The wild type of this network will produce spike skin cells because S will activate P, which will then block I, so that I cannot block K, and then K can activate E, which will activate the spike gene. If there's a mutation with P, the network will not produce spike cells because P will not repress I, and I will then be able to repress K, which is needed to activate the spike gene. A mutation with I will produce spikes because I will not be able to repress K. A mutation with I and E will not produce spike gene in cells because I will not repress K, but the mutation to E will stop it from activating the spike gene. A mutation with S and K will not produce a spike gene because the mutation to S will stop P from activating and thus repressing I, but also mutant K will not be able to activate E. A type of regulation that could affect the expression of the spike gene in skin cells would be the methylation of DNA. When a methyl group binds to DNA, it causes that DNA strand to coil tighter around histones than it already was, making it much harder to read. This makes some genes on the strand silent due to them being inaccessible to being transcribed. This silencing does not happen with every DNA strand, so with the strands it does occur, there will be genes not expressed, and with the strands it doesn't occur on, all the genes will be expressed. In the genes that are not expressed due to the methylation, the developmental network is still in place and occurring, but the outcome of whether or not that gene will be expressed is not seen due to the tight coiling of the DNA. Thank you for watching, and I hope you all have a great day and have enjoyed this video. My sources for this video will be linked in the description below. Thank you.